We specialize in super resolution techniques, which are light microscopy techniques, which are able to see structures underneath the diffraction limit, so smaller than about 250 nanometers. They can be used for a really wide spectrum of things. So in the academic setting, obviously it's mostly like disease research and things like that. So across cancer research, neurodegenerative disease, that kind of thing. But really, it's really just about the size of the things that people need to be able to see. It could be applied to almost anything. Our most popular technology is our STED microscope, which is stimulated emission depletion. And that one can allow us to see structures down to about 50 nanometers, or we can also do it in 3D down to about 70 nanometers in all three dimensions. And we can do that in multiple colors. We also have a system that we use for single molecule localization microscopy, and that can get down technically to about 10 nanometers in resolution. We also do a technique called fluorescence lifetime imaging, which isn't in itself a super resolution technique, but we combine it with FRET, which is Forster resonance energy transfer, and that allows you to see interactions between proteins that are occurring at sub 10 nanometer distances. So even though we're not getting a spatially super resolved image, we're looking at interactions which are happening at this super resolution scale. The advantage of these technologies is that we're keeping the benefits that you would have with electron microscopy in terms of really pushing the boundaries of the resolution, but we're then allowing for multicolor labeling of specific proteins or structures, and we can do it in live cells, so we can actually look at the dynamics of these structures. And then finally, we have a system which isn't technically an optical super resolution system, but does still allow you to reach very, very high resolutions, and that's an atomic force microscope. And we can use that either to get very high resolution topographical information about a sample, or we can also use it for doing force spectroscopy, which can give us an idea about the stiffness of different parts of a sample. And we typically use this for biological samples because the system that we have is specifically designed as a bio-AFM, but it's also a technique that's quite commonly used on materials and those kinds of things. And what I do is train researchers who come here from different universities around Scotland who want to use these technologies for their research. The majority of our users are academics, but we do some work with industrial partners. And yeah, it's really just about finding, I guess, the industries where they will actually need to be able to see things that are small enough that they wouldn't be able to do it using traditional technologies. As far as I'm aware, there's nowhere else that has this concentration of these technologies in one place with the expertise in all of them.